From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 93, recorded on August 18, 2023. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Hello, everyone from semi-tropical LD Lab Studios over here.
Is it warm there?
It's humid, so we've got the almost the preview of Hurricane Hillary coming our direction, at least in a regional sense. So I think it's going to hit Southern California, Baja area, or it is right now. I don't know the exact track, but we're anticipated to get some remnants, I think, in these parts. Although I guess it can still steer various directions. Might be more Oregon, Idaho, etc. But we'll see. You can already feel the humidity here, though. Yeah.
We're at 26 here. It's not too warm. It's,
not bad at all. Yep.
Quite low, yeah. And it's been mild. We don't have that hot weather that you guys are all having. The rest of the country.
Yeah.
I don't know why.
We've kind of been on the edge of it in this sort of opening chapter of the new abnormal, as they say. I feel like every time we connect every month, there's a different sort of weather event to talk about, and try to sort of manage through somehow. But
welcome everybody to an unusual day for TWiEVO. It's the only one we could get this week.
Yeah, you've got a full podcasting menu, I believe. You've got a TWiV on tap as well?
Yeah, we got a TWiV in a couple of hours today. Yeah, I did 3 yesterday. Man, that was a tough one. What did I do? I did a TWiV clinical update. We did TWIP. And what the heck was before TWIP? Why can't I remember? TWIM. It was TWIM. Yeah. And then today, too. I want to welcome our moderators. Barb Mac UK, thank you. Andrew is here.
Awesome. Welcome.
I believe those are the only 2 mods. They're probably in, I know Barb Mac, let's see, it's 1 o'clock, so it's like 7 p.m. there. It's not bad.
Not too bad.
Thanks for coming. Who else we have here? Let's see. Nicola is from Italy. 30 C and 62% humidity. Claire is from the UK.
Welcome.
Let's see. Lise is from Columbus, Ohio.
Nice.
Let's see. Helsinki. M.E. Wheeler is from Helsinki. I just got invited to a meeting in Finland next year. I'm looking forward to that next June.
Awesome. That sounds amazing.
I like Finland. Finland's
a lot of fun. I've never been. I have a electric sauna heater on order from Finland. I think it's on a freight ship right now or something like that.
That's funny.
I'm a big fan.
Peter, I don't know where Peter is from. Do you know what language that is? Is that like New Zealand or something?
I think we're coming from New Zealand over there.
All right.
You'll have to confirm that for us, Peter.
Stefan is from the Netherlands. Jeff is from Santa Barbara.
Some soon to be wet weather. That hurricane is bearing down, I think, almost directly on Santa Barbara potentially.
Kathleen is from the Jersey Shore. Frank is from Santa Barbara. Fields of Green is from Salinas, California. Chris is in Edinburgh. Environmental is in the southern tier of New York. And look, AZ is from KSA. AZ has been communicating with me.
Oh, great. Welcome.
I have a box that AZ sent from for Daniel. It's right behind me here at the incubator. Oh, Les, welcome. Nice. Thanks. Another moderator. Thanks, Les.
Good to see you.
California. Environmental is Sandy. Sorry, I know that wasn't your actual name, but I didn't know. Yes, Peter is New Zealand. Peter and Andrew are both from New Zealand. And
awesome.
Pete is from London. Yeah, I just put up the schedule for this yesterday. I totally forgot about it because it's kind of an unusual day. We don't really ever do TWiEVO on Fridays, right, Nels?
No, we're throwing a little bit of a TWiEVO curveball here. This is how we figure out who our true fans are, the ones who will show up at a drop of a dime, moment's
Yeah,
notice,
exactly
strange
right.
time. So this is the true aficionados here. Thank you all for coming. Great to be together.
Yeah.
Yeah,
So we have about 34 people. Yeah, that's usual for TWiEVO.
yeah, it's good.
So thanks everybody. We have an interesting paper today that I think you'll find is pretty straightforward, but has a lot of implications, don't you think, Nels?
I do, and I'm grateful to you, Vincent, for pulling this one up. So I had missed it actually. I was looking at there's a companion paper, I believe. So we're going to do a deep dive into synthetic biology as it intersects, interestingly, with experimental evolution today. And so there was sort of two papers. This one was hiding kind of in plain sight for me, at least. And so I'm really glad you pulled it up and said, hey, let's do this one for TWiEVO.
That's great.
So the paper title, simple title, which is a good sign right out of the gate so that it might be clearly communicated, is Evolution of a Minimal Cell. And this builds off of, decades of work, actually. And the idea, as recombinant technology has gotten better and better, the idea that you can actually synthesize an entire genome and launch it in the cytoplasm as a new replicating critter. And so, yeah, thanks for putting this up. So this is an article. It's open access for anyone who wants to do a deeper dive, freely available at nature.com. And group here from sort of all points. The heart of the work is coming out of Indiana University. This is Jay Lennon's lab in the Department of Biology there. And first author, Roy Moger-Riescher, sort of, I think, kind of leading the charge here. Then with collaborators from San Diego, the Novartis gene therapy wing, kind of hinting at some of the synthetic biology, some of the technology here, perhaps. And then a group dedicated at the National Institute of Science and Technology in Synthetic Biology in Brazil, also part of the crew of the U.S. Geological Survey, Wetland and Aquatic Research Center in Lafayette, Louisiana. And then the second to last author is a fellow called Mike Lynch, who was at Indiana University and is now at Arizona State University in Tempe, doing really interesting stuff in the field of evolutionary cell biology, area that I'm sort of would consider myself a practitioner in that sort of broad field, taking evolutionary ideas and really trying to breathe life into them with experiments. So great crew here. And as I kind of was hinting at, I think an interesting collision between synthetic biology and experimental evolution. So Vincent, do you follow the synthetic biology field?
I don't really, but I did this paper, not this paper, but the synthetic mycoplasma that we'll talk about was done quite a few years ago.
Mm-hmm.
And I remember reading the paper, and I swear I did something. I either blogged about it or did a podcast. I can't remember. I can't find it.
Yeah.
And so, yeah, I remember the initial paper, but I stopped following it. And then this one popped up recently in my feed, and it looked really interesting. So, but I don't. And do you follow it at all? You do?
Yeah, not super closely. So I actually remember that paper, that first sort of splash, and I think it was around 2010 when, species here is Mycoplasma mycoides, and picked in part because it's such a small genome as a natural creature, natural bacterium. And so this is Craig Venter and others at the JCVI, the J. Craig Venter Institute. And at the time, so I remember reading it and thinking at the time, I was, to be honest with you, Vincent, a little bit underwhelmed. So what they did, which is and don't get me wrong, it's a massive technical feat. The technology here is pretty wild, and credit to this group for really pioneering the ability to synthesize, in this case, 1 million base pairs of DNA to kind of get that together through a combination of plasmids and stitching and everything else. And then taking that genome, putting it into the cytoplasm, or I guess they describe it as the bacterial shell of another mycoplasma species. And then sort of the magic happens where it's sort of like plug and play. That genome, encodes the proteins that then are translated to start to put into motion the biology that it takes to be a self-replicating critter. I think where I was maybe a little underwhelmed conceptually is that nature was doing it a lot better back then. It was just like they kind of copied nature and then did that trick of putting it back into a slight. But I think actually, honestly, with a little bit of hindsight now, whatever, 15 years on, maybe I become more and more, interested or impressed, not only with the technological feat, but how it kind of provokes the imagination a little bit to think about, well, what can you do with this? Or how does this thing evolve? Which will really be the topic of our discussion, our conversation on TWiEVO 93.
Mm-hmm. I think it has some interesting ramifications here. And so this kind of organism is useful, certainly for looking at evolutionary questions. But as they say, there are other applications of making minimal organisms as well, biotechnology applications and so forth. I see where the Ventnor Institute would be interested in it, because that's sort of what they do, right?
Oh yeah, absolutely. So a lot of the early work on just pioneering genomics technology, racing, like the Human Genome Project, started getting into the race.
Yeah.
So, you could make the argument that there's certainly not small egos at play when we're talking about Craig Venter and his sort of pursuit of scientific stuff. But that also, breathes some, on the positive note, that breathes some excitement and energy into it too. It sort of personalizes it in some ways as well. So, but let's talk about mycoplasma for a moment, sort of the star or the hero of this story. So this is a bacteria in the genus, maybe most well known among molecular biologists for contaminating tissue culture experiments. We do we do a lot of work sort of monitoring, hoping that mycoplasma doesn't show up in our cell culture media or in our cell samples. But obviously that wasn't the original niche of this bacteria. So this species in particular, Mycoides, you find in ruminants like cows, I think water buffalo, other ungulates ruminants, and it can actually cause pneumonia-like symptoms. And so there is, a real also kind of agricultural or environmental issue here. But outside of that part of the story, for this one, the reason it was so attractive is that it's such a small genome for free-living bacteria, even among bacteria, coming in at roughly a megabase, I think something like 900 genes, really streamlined relative to most of free-living life that we've cataloged over hundreds of years. And so I think that was,
by the way, the original complement of the free-living guy was 900, right?
Exactly, 900 genes. And so in about a million base pairs. And so they had just enough kind of reach, even 15 years ago, to synthesize, to stitch together. They didn't do it all in one go, if I'm remembering correctly, but were able to sort of cobble that together and reach that number. And then it also, I think, in addition to sort of the practicality of targeting this species of bacteria, it also raises some interesting questions, evolutionary questions about sort of minimal genomes. How streamlined can you be? And then how do you evolve? Or does that influence some of the key parameters of evolution, things like mutation rate, the nature of mutations, and things like that? And so I think, echoing on your point, Vincent, about, all of the sort of increasing number of biotech applications, like one of the massive open questions is, like, let's say that you engineer a bacteria that's really good at metabolizing oil, or, breaking up oil at an oil spill. But if you've kind of streamlined it so much, or it has kind of evolutionary properties where it's unstable, you might put all of this time and energy investment into launching this right as you hope to meet kind of in that case, like an environmental disaster or whatever it is. And then if it sort of hits a evolutionary dead end and just, like you go from a million cells down to zero, like then you sort of have done all of this for nothing. And so I think that's where, definitely this, from a, maybe a applied standpoint, where you're learning about what happens as you, in this case, use experimental evolution, the repeated sort of culturing or passaging of these bacteria over time, to then compare the sort of adaptive potential and the mechanisms that these things, like what this means for fitness for that in that population. And then it also, I think, just from a more academic standpoint, there's a lot of interesting questions just about, kind of curiosity driven questions about, if you build this thing, what's going to go on? And so, please go ahead.
I just want to mention, they call this genome streamlining.
Yeah, that's pretty good. Yeah.
And originally, so we should point out that a lot of the genes they took out, they don't know what they do.
Exactly. And so so now I'm glad you brought this up, because so this takes us from sort of that 2010, where I'm claiming I was underwhelmed, to 2016 or so, where they've actually developed this into version number 3. And I don't know what happened to version 2, to be honest with you. Maybe that was sort of left on the cutting room floor, the genetics cutting room floor or something. But but at least version 3, what they've done is they've moved from 900 genes down to something like 400 genes. And exactly like you're saying, they don't know. The goal was not to like know what all of the genes do and then do sort of a logic based, let's take these things out. It was more, let's see what we can take away and let's see what we can get away with, basically, right? What can we take away?
Sure.
And the thing is still able to replicate. And so that's where the streamlining happens. And it's pretty wild, right? So now they've gotten it down to like less than half the original genes there, and yet it still replicates. And so they can make the claim, which is really cool, right? So and that's what they mean by, that's how they're defining here, minimal cell. And we could quibble, I mean, is this really, could you do a little more streamlining, et cetera? That's a good reason to do some experimental evolution. But the smallest genome of any known organism that can be grown in lab culture is what they've come up with here. Right. And that's a pretty, that's actually,
And remember, this is in a medium, in a lab. It's a rich medium probably, and it's not competitive. Whereas out in the wild, maybe some of those genes would be needed, right? And so it's a different question. So
I agree with you. Yeah, and it also sort of that ability to live independently, I think, is actually an important point to take a minute to sort of weigh. And there's all kinds of streamlined genomes that you find in nature, but that usually is happening in a process of like the process of becoming a symbiont. So, we've talked about this on TWiEVO over the episodes, which is that given enough time and a close association, something that arrives in kind of a long-running interface with a host might actually become sort of part of that biology. Of course, the mitochondria in each of our cells are sort of the quintessential example of this. And so in that case, what happens quite regularly is this process of genome streamlining. But there, you don't have to be able to replicate independently in lab culture or anywhere. You've now sort of hitched a ride with the host, genetically speaking, where a lot of the functions needed are encoded in the host genome, in the nucleus, if it's a eukaryote. And so that also, I think this kind of carves out an interesting sort of biological space, or a place to experiment where it's sort of streamlining in another context, which is still maintaining the constraint of living independently. And so, kind of one of the only games in town, as far as I know, in terms of thinking a little bit about that.
I wonder how the engineering went, Nels, if they just took out a lot of genes, or if they had to do it stepwise, and there were some failures and so forth. I'm not remembering the original paper. I'm not sure they gave us a lot of insight into that.
They haven't, but so that's where I would, if our listeners, all of us, are interested, this companion paper, I think they do a pretty good job of laying out how did they come up with this version 3, and then kind of kicking the tires, test driving it. And a combination both, I think, of sort of the classic genetical, let's just mess around with this, take some things away, sort of experiment with it that way. But at the same time, looking at what are the predicted open reading frames or known open reading frames, and where there are functions. And as you pointed out, that's like actually still, I think, maybe less than half of the gene functions are known. But, making some predictions on essentiality there, or even through sort of like, distant homology and things like that. So that can guide it a little bit. I think it's sort of a handcrafted approach to genome streamlining in this case. But yeah, that other paper I think takes a pretty in depth, if I'm remembering it. So I was, when we were kind of getting ready for this episode, or maybe the last one, I looked at that other paper and was almost going to suggest that one, not knowing that there was this one focused on evolution. And as I got into it, I said, oh, that's probably not a TWiEVO paper. It's just really kind of a long, detailed look at how this sort of synthetic biology works. And so, not that it's not interesting, it is, but it sort of weighs on some of those other questions out there.
Which paper is that one, Nels? Do you know of?
Yeah, so I think it might be in the same issue, potentially at Nature. I'm not 100% sure about that. It came out about the same time, maybe a month and a half ago. Venter would be an author on that one. Okay. We'll pull it up here one way or another before our time is up. But for this study, what they did was they took JCVI -SYN 3b, which is what they've named this Mycoplasma mycoides synthetic version. And the researchers actually grow this two different ways over time, sort of in this experimental evolution framework in replicates, and want to compare if they passage for 2,000 or so generations, and that takes on the sort of when you account for the replication speed in these populations and the rich media that they're culturing them in, that takes between 20 to 36 weeks. So, a semester or so of time to really see these things change and right before your eyes. And there's two flavors of experimental evolution they're doing here. And these are sort of important, I think, markers to lay down, or definitions or concepts for all of our evolution curious listeners. The first is mutation accumulation. So here the goal is to really just get a sense of sort of the population genetic parameters. What's the mutation rate? What kinds of mutations are accumulating over time? And so under that scenario, what you want to do is take away as much of the selective pressure as possible to sort of allow just the natural biological process to take place, so that you can make calculations by comparing the genome sort of before, during, and after that course of mutation accumulation. And so the real sort of trick here, the secret sauce of doing mutation accumulation, is to bottleneck the populations at each passage. So you grow them up a little bit, and then you just take a couple of marbles out of the bag, so to speak, put that into culture, and keep it going and going and going. And the reason you want to do that, so that mimics what sort of the property of genetic drift, which is they're not, as they're growing and the population is becoming more heterogeneous, genetically speaking, they're not competing against each other, because you're just imposing, it's like by chance, you're the ones who win into the next generation. And so that's genetic drift. And it's a little bit of a subtle concept, in the sense that doesn't mean everything is random and nothing is happening. You still need, obviously, to have all of your essential functions in place in order to replicate going forward. And so that's kind of a key distinction to make. But really what they're doing is trying to take away competition, let these things replicate, and then see how many mutations sort of accumulate over time. Mm-hmm. So that's in contrast, and we'll dig in a little bit to what they found. Comparing, again, that 900 genes version 1 to the 400 or 473 gene minimal cell at roughly half the genome size. And does it accumulate mutations differently? The second approach here with experimental evolution, this is one that we do in my lab. A lot of people do. If you're curious about the sort of how fitness is impacted through selection or competition or various things like this, we did this recently, Vincent, on algae in the mason jars.
That's right. Where last one, yeah.
Last one, yeah. So where they were specifically trying to set up competitions, either within or between species here of algae or predators or competitors, et cetera. So there you want to purposefully put on selective pressure. In this case, it's sort of a subtle one. They just let them grow up, and they don't bottleneck as much. So the ones that accumulate mutations that might outcompete the others around them will sort of be the ones that sort of propagate or go forward. And then with that as an approach, you can then say, okay, does it grow faster? Does it grow better? Is it adapting? How does it compete against other ones? And then look at some of the other parameters. And then what are the mutations that were, adaptive or beneficial in that population? Okay, so they do both. And they point out at the kind of outset here, I think it's kind of a fun quote that I thought I would put on the table here, which is given this is these bacteria, the mycoplasma, have really high mutation rates. I think, some of the highest per generation that you see among free living organisms. So the mutation rate is it doesn't sound super impressive, 3 times 10 to the minus eighth nucleotides per generation. That's when you think about how fast generations go and how big the populations are, with population sizes exceeding 10 million individuals in these experiments. A new mutation would hit every nucleotide in the genome more than 250 times during 2000 generations of experimental evolution. So this still, I have to say, like, constantly blows me away, is when you're doing, this sort of test tube or petri dish evolution, that within a semester's worth of time, every position in the genome is seeing a mutation 250 times, right? It's almost like it can feel like magic when you're doing this and all of a sudden, oh, here's a mutation that swept through the population. But kind of under the hood here are like tons of failed experiments.
Yep.
Even in this like small, very controlled scenario. Now, go out into nature. Think about SARS-2. How big are these virus populations? How much is that sampling? And that we're just sort of almost like right under our noses is this massive churn of genetic diversification, which I think we massively underestimate at our own peril. And this has caused some political dust-ups, to say the least, recently.
No,
as well.
it's a very important point, because to get a virus randomly that can infect people pretty efficiently, that's, a lot of random mutation going on out there that we're not even seeing, right? And people don't think that. They don't realize that that's all it takes. And it happens in those bats. There are lots and lots of bat hosts, and there's a lot of replication cycles per bat per day. And it just generates this enormous diversity from which, given the right contact with a host, you can select out the right virus to spread. And so it doesn't require any laboratory manipulation whatsoever. And so this is really important to emphasize this. I don't think people really get that out there's an enormous diversity, and that's what gives rise to spillovers. But the fact is, we don't have many that give rise to pandemics, right? It's pretty rare in the scheme of things. So
exactly,
most of them fail. They don't work. They're not the right. So that's why when one like SARS-CoV-2 does what it did, it's really an incredibly rare event. But that just tells you how many mutations and mutated genomes there are out there to give rise to that.
Yeah, exactly. Yep. No, and I think the human brain is not, like myself, my human brain included, is not wired to sort of grapple with those numbers. And so I think it gets, we just kind of push that to the margins pretty sort of naturally. And it's a big, at our own peril. Like that's where the game is being played. That's what keeps me up at night, is not what's happening in the laboratories so much as what's happening in nature in terms of the risks there on a natural basis. Anyway.
That's the problem, is that nobody sees it, right? Very few people see that.
It's invisible.
It's a theoretical thing. It's happening. And so if you can't see things, whereas we can see there's a lab somewhere working on coronaviruses, it makes sense to people. But the other thing I want to point out, Nels, is that when you make so many changes at each base in a genome, that many of them don't work. Right? They don't work. They break it. They break the machine, and many of them have no effect whatsoever. So you have to keep that in mind, that not every change is going to have some consequence.
Exactly right. Yeah. And so here, even with this sort of relatively modest experiment, because they've set it up and know all these parameters, they have some real control over these critters, over this bacteria, the synthetically generated one. They can look at these parameters and say at every position, we've got 250 changes over even the course of this. So that's taking the invisible and measuring it sort of in the course of this, using genetic techniques and tools. Okay, so what do they see? So the mutation rates are high, as they point out, for bacteria. Not high compared to viruses like RNA viruses, by the way, but high for bacteria. And the types of mutations turn out to be really similar between both. So the comparison they're drawing, right? Yeah, here exactly is the non-minimal.
Says it all, right?
Figure says it all. So we've got, panel A at the top, non-minimal. That's that 900 gene, the sort of version 1.0 versus minimal where they've cut it in half. And so those little violin plots, as they're called, each dot is they're measuring a mutation rate as they're sampling through the populations. And you can kind of see mutation rates are overlapping. The dots are basically on the y-axis, end up kind of in the same distribution.
Nels, how would you measure a mutation rate? Would you do genome sequencing, basically?
Yeah, exactly right. Yeah, and they do it. You can kind of dig in. I think it's pretty well written, or the paper itself is very well written. The methods are pretty well written, and have a lot of those details of how they're kind of calculating these rates sort of at different intervals during that 20 to 36 weeks of mutation accumulation. And so they're actually not taking, I don't think in these cases, for these calculations, they're not taking the entire genome at every position, but sort of downsampling a little bit, but getting a sense. And then they can go through, and we're seeing in panels B and C, sort of digging in on the flavor of the mutations and comparing that between whether you have a streamlined or, original OG version of the genome. So insertions, roughly the same. They're pretty rare. Deletions, roughly the same, a little more frequent. And then SNM, single nucleotide mutations, roughly the same, actually, maybe a little bit of a reduction there in minimal. Where they start to see a little bit of a difference is in some of the when you now go to the whole category of single nucleotide mutations and break that down even farther. So do A's and T's go to G's and C's, the reverse, the A to T bias, as it's called as well. They see two, I'd say, interesting differences. One is a more of a lack of the so-called neutral mutations in the minimal, in the middle there, and then also a flip-flop in AT bias. And so they actually, they kind of to look through what's going on here. So I think the big take-home message here is actually very little overall difference, and even pretty subtle difference in the kind of mutational basis. But they note that when they did that streamlining, they took away a gene called UNG, which is actually involved in some of the repair processes. And so that probably, without that in place, because you do repair differently in the minimal gene, just sort of mechanistically, that might account for those differences. But it doesn't seem to have like a huge impact, and the mutation rate overall is about the same. And I think they've taken away something like 7, repair pathways, which is, a big investment for some of these things. It remains pretty stable. And I guess maybe one of the take homes here is that, the mutation rate is sort of driven in large part by the polymerases, the DNA polymerases at work that are doing the copying here. That's sort of cooked in to the enzymology there of replicating.
I guess they're surprised to see that they took out all those repair pathways and mutation rate doesn't change, huh? Right?
I think that was a surprise. Like, it's one of those things, you do the experiment and maybe at first you're a little bit disappointed, but then at the same time, it's like, wow, this thing is actually more kind of stable or robust as an evolving system than you might have first thought, with all of the, with literally cutting this thing in half and still just maintaining what they're defining as a minimal cell. Okay, so then they move on to that second flavor. And I think there's one sort of, interesting take home to me, and we'll get to that in a little bit, a little bit later in Figure 4. It relates to the morphology of these things. But they now move into the experimental evolution, where they're actually asking about fitness. And so instead of doing all of that bottlenecking where it's marbles from the bag, they want these things to compete a little bit. And so it's almost the same experiment. It's just from passage to passage. So as the cells grow up, right, in a test tube or petri dish or mason jar, according to our last episode, what the cells will reach, they'll use all those nutrients, or they'll replicate so much that there's no more space physically to replicate more, and then move into stationary phase. So instead of taking just a few cells as they're kind of coming to the end of their ability to replicate in that first vessel, now they're actually taking not the entire thing, because otherwise you'd need a bigger and bigger vessel. The mason jar would become sort of a whiskey bottle, and then that would become a keg or something like that. Here, what they're doing is they're still sampling down, but there's enough cells coming through that they can kind of compete against each other in real time. And so that allows them to then think about adaptation, to think about fitness. And so here they're defining fitness in a couple of different ways. One as a maximum growth rate. And then they also do some competition experiments in this case with the ancestors, the sort of cells or the population at the beginning of the experiment, which are pretty homogeneous. They've literally launched this as a synthetic genome. And then comparing that to the ones that have gone through the 2,000 generations of experimental evolution. So a couple of take homes here from the maximum growth rate. So those minimal cells, you can't just like take away half the genome, half the genes or so, and expect everything's going to be perfect for those cells. So they start at a much slower growth rate. And by that definition, they have lower fitness, but they improved linearly over the course of the experiment. And so, and sort of on the same linear basis, almost in parallel, to the original version 1, where it's not as streamlined. It has the 900 genes or so. Where it gets, I think, even a little bit more interesting here is actually in the competition fitness. And so, again, you take like a pretty big hit in the ability to compete. So they're only like half. So the competitive index here is 0.5. If you compare them to the non-minimal. But here's what's cool. So they basically, in these competition experiments, they compare the non-minimal to non-minimal, and you set that at 1, right? So at the beginning of the experiment, as ancestors, they compete equally, which makes sense. They're the same population. And then when you do that evolution of the non-minimal and then compete them against the ancestor, they're better. The evolved ones are better. They outcompete. But what happened on the minimal case is they started lower. They basically approach the ability to compete with the non-minimal at the beginning, but they do that at a faster clip. The sort of rate of competition increases. And so they're actually adapting a bit better or faster in sort of this competitive index than are the non-minimals. They still don't approach so the non-minimals evolved 2,000 generations still compete better against the ancestor non-minimals than the evolved minimals. But it's like sort of tortoise in the hare maybe here. The tortoise is slowly sort of catching up in competitive index. And if you extend that experiment maybe for another, 8,000 generations or something, do it for a year or two, and maybe this is happening even as we speak. I think it'll be interesting to see what happens. Do they plateau? Do they continue on that steep curve? Some of the long-term evolution experiments that Rich Lenski is famous for, now up to probably 80,000 generations or something like that. In that case, you often see a lot of the fitness hitting these plateaus for a while. And then in some cases, in some of the replicates, something happens. And then there's sort of a new sort of evolutionary chase that can unfold. But here it doesn't look like it's
let me just clarify. So
please.
Basically the minimized genome is less fit in these assays, like 57% reduction in the maximum growth rate, right? And then as they go through these 2,000 passages, it slowly recovers fitness. Its growth rate increases at the end 68%, right? So that it's getting better just by passage, which I guess makes sense because you're just growing it and you're selecting for mutations that improve growth rate, which makes, I guess that's, there's no selection pressure here in Nels. It's just growing. So I guess at the end of each cycle, you pick the ones that are growing better, essentially, and the slow guys get left behind, right?
That's right. And so here, it's kind of a subtle difference, but so you're exactly right. You're not putting, so like, in a lot of experimental evolution, you might put the selective pressure you might put on is to say, okay, for a virus, for example, here's a host that you barely recognize or can barely replicate under, figure out how to replicate better. That's a really strong selective pressure. Here, as you're pointing out, right, they're in rich media. There's no predators there trying to eat them. They don't have to run away or anything like that. Life is pretty good. And so the only difference in the parameter is how many cells come through each bottleneck. And in contrast to the mutation accumulation, there's enough genetic diversity coming through each passage that they're sort of still they're competing against each other. You don't start as a new homogeneous population every time, or you don't have to compete because you're identical, right? And so, even in that, so no selection, or you might say like really low selection, just based on competition with your heterogeneous sort of, genetic sort of close siblings almost. That's driving the evolutionary process. This also, really important, I think, from a practical standpoint, getting again to some of these issues of if you spend a lot of time bioengineering something for, to make a better beer or whatever it is, you can probably expect that this thing is going to start out of the gates pretty weak in its ability to replicate. And so this is sort of like a nice encouraging that it can kind of get back to the starting line, basically, with its sort of natural cousin, or well, it's still synthetic, but its cousin that's closer to the natural thing to begin with. It's and that's, I think going into the study, I think this is some of the novelty of this, and the reason that I found it really exciting to kind of read through it is, and there could be other cases where you just like put your synthetic biology or your genetic engineering is so far off the rails that you can't recover back to something approaching sort of a natural competitive index or growth rate. And so, but in this case, it was okay. And they did quite a bit of tinkering. They cut this thing in half. And so that's a pretty major hit. And yet this thing keeps coming back. Maybe a hint at the robustness of evolution. If you can grow a little bit, before too long, you can grow pretty well.
And as this passage happens and the growth rate is improving, it also improves in a competition assay, as you mentioned. And so that's they take at different points and do a competition, and so that's clearly allowing this to get better. And in the end, I think it's almost as good as all the fitness lost was recovered during 300 days of passaging.
Exactly.
It's amazing.
Exactly. I love that. Yeah, I love that. And then, but Vincent, it gets even better because these are simple critters, you can you can sequence the genomes at various time points and consider the genetic basis of adaptation, not only measuring that they are better, but to ask why, or, what is it that's different, what that has changed that allows them to compete better. And so here in this case, they find they identify lots of candidate pathways. This is a little bit challenging with the bacteria with such a high mutation rate, even over 300 days or so. The number of mutations across that genome is pretty vast, even if the genome is less than a megabase. And so really difficult to take, and a lot of those mutations, as you pointed out before, aren't going to have any impact at all. They're not going to be so deleterious, because if they were, then those ones wouldn't show up. They wouldn't be able to compete at all. But it's discerning that difference between, beneficial, the rarish ones that are beneficial, and then the ones that sort of are just passengers, don't really matter. And that's a mess already, even in a small course of evolution, given that you're hitting each position 250 times with the mutation. That's a lot of mutations. They're not all there at the end of the experiment, but even the ones that have sort of come through, whether neutral or slightly deleterious, neutral, et cetera. Yep. So they kind of do a little handcrafting, I'd say. They kind of pull this down to candidate pathways and look for the overrepresentation of what appears to be a non-neutral mutation, and find 15 or so, both in the non-minimal populations after 2,000 generations, and then also in the minimal ones after 2,000 generations.
Are they the same changes in both, Nels?
They're different. Yeah, great question. So they're different. Yeah, and that sort of, from a experimental evolution standpoint, that gives you the sense that there's like a lot of ways to get better. You don't just have there's not kind of one mutational path to go forward. There's probably all kinds of solutions, and they don't go really into the replicates, like how much they do between non-minimal and minimal, and say these are quite different. They don't go so much into the replicates and say how much, did you follow the same path or different paths. Yeah. My sense was it was pretty different. So there's kind of flexibility in the ability to compete here. And then, because you've got such great genetic control, the ability to manipulate these genomes, they go back and, at least in a couple of cases, use CRISPR editing to go to the non-evolved, the parental sequence at the beginning, make that change that they're suggesting might have been important in that adaptive process, the ability to compete better, and then test those for fitness. And so the one they kind of zoom in on and really focus on, including kind of into the last turn of the paper, is a gene called FTSZ. And this is a GTPase that's involved in membrane pinching for cell division. And so obviously when you're talking about replication, how you pinch from one cell into two to four, 4 to 8 in exponential growth, how that action is happening is going to be important. And they can see that by making that mutation, And I don't think they really drill into the entire molecular mechanism here, but they're able to recapitulate some of that adaptation just with that single mutation, sort of in the sea of many. And so that's not to say that's the only mutation. I think they recover like 60% of the fitness from that one, which is quite large for a single mutation. But it's the combination of all these things. There's another complexity here that they mention but barely drill into, which is epistasis, the combination of mutations. And so even if you've hit 250 mutations at every position, as you start to get into, well, what about these two mutations? If they showed up together, they've still only kind of scratched the surface of evolutionary potential, adaptive potential, when you hit these jackpots of like 3 or 4 mutations. And again, we see some of that echoing through things like virus evolution, SARS-2 evolution, where it might be 30 changes to the spike protein that have never been seen before, where epistasis is at play. The combinations of mutations are starting to be selected on as you increase the population size, the time, etc. So the other genes that they sort of point to are the other genetic basis of adaptation here. They went through 15 or so of these pathways, and also were curious which ones might be exclusive to the minimal genomes versus the non-minimal. And they even, I think they kind of balanced their argument a little bit here. They said they proposed one that they thought for biological reasons might allow the minimals, to sort of deal with getting nutrients across the membrane, since some of the things they eliminated were the ability to make the metabolites or nutrients that the bacteria needed. That maybe what would happen is they would get more efficient at just grabbing that from the rich media. They didn't actually see that. They did see maybe like indirectly something going on here, which was some genes involved in lipid metabolism. And that those changes again appeared to allow the minimal cell to use the nutrients better, or to have a lipid composition that allowed it to replicate or compete, but a different pathway. So even that, at the end of the day, you might end up with sort of the same growth rate, similar growth rates, et cetera. It's for different reasons, different biological reasons. And again, that'll be really important as you get into the weeds of the kind of, sort of synthetic biology that you might be practicing, in order to start to get the concepts of what are the sort of things that allow these things to grow, or, things to keep an eye on, where you might be able to sort of jumpstart the growth ability of a different bacteria that, that someone might be synthesizing for an applied reason.
What's interesting, Nels, is that the genes that seem to make a difference are not the ones in biosynthesis, but the lipid genes that you were talking about there. They seem to be okay in terms of metabolism, right? Maybe because of the rich medium that they're grown in, right?
Yeah, and maybe that, like already the ability to get those metabolites across the membrane isn't in doubt, that the minimal set of genes sort of like gave you that baseline as a cell to be able to do that. And I think they thought that there could be some more biological optimization there that maybe wasn't actually so necessary. Yeah, that's right. Yeah. So then, the last kind of turn here, the paper is thinking about just looking at these things. So far they've grown them, they've measured how many they are, they've dissected out the genomes, but we haven't done maybe the most simple of experiments, which is to grab a microscope and just look at the cells and say, what do they look like before, during, and after the experiment. And
that's something that Petra Levin always says. She's on TWiEVO, and she says, you always have to look at, she works on Bacillus subtilis. She says, you always have to look at your cells. Sometimes you find really interesting things when you just look at them under a microscope.
Yeah, that's right. And sometimes, maybe that's my other small quarrel here with synthetic biology too, is that you get sort of caught up in these like fancy genomic techniques and genome engineering that sometimes you forget the sort of easy ones. But they didn't forget here. The evolutionists here didn't forget this, population genetics, et cetera. They looked at these guys under the microscope, and there's a good reason to as well. And actually, we talked about this a little bit on the last TWiEVO, which is some of this, right? The cells, like what happens to populations under selection in terms of cell size, even animal size and population numbers. So this gets, we haven't done too deep of a dive into the population genetics here, and I don't think we will, but we went a little bit into this last TWiEVO. And so that brings up the question of cell size or even morphology. And here, in contrast, I would say, to some of the, like the mutation, sort of the flavors of the mutations, et cetera, were kind of roughly the same. Here they see a really cool difference. I don't think that they actually explain the basis of it, but it's a pretty striking difference
Mm-hmm.
in cell size, right? So
Yeah.
yeah, here we go. So those minimal cells in that graph in red in the dots, so the square is the average, and each dot is looking at some of these cells. These are electron micrographs, and you can actually see a little collection of them on the right there, are smaller, both at the beginning of the experiment as
Yeah.
ancestors are roughly the same size. There's some overlap there, obviously, between the blue and the red dots. But over that course of where they've gotten better, they're able to compete. They really haven't changed in size. And that's different. They also look a little bit, I don't know, you can see the kind of those threads between them. They look a little more rough kind of on the
Mm.
membrane there. But that's in contrast to the non-minimal cells. So they started out a little bit bigger. And we're measuring cell diameter on the y-axis. And then as you get to that evolved population, you can actually see that they've increased in size quite a bit. And that follows some of that life history theory in population genetics that we talked about on last TWiEVO. But the minimals, they didn't get so much bigger. And they connect this to that mutation in FTSZ that they saw over the course of the experimental evolution, and they did the CRISPR editing to put back. And so what's wild here is that when they then go to the ancestral population, whether it's the non-minimal or the minimal, they put that FTSZ mutation into the ancestor, so it doesn't have the other changes that accrued over 2,000 generations, but they get opposite effects in the minimal and the non-minimal cells. It's beneficial in both cases, and they can recover some of that fitness. But in the minimal cells, just putting that in, the cells get bigger. Whereas in the non-minimals, they just put that in, the cells get bigger. In the minimals, they put it in and they stay roughly the same size.
Yeah.
So
Yeah.
that's a really cool difference here, actually, in some of the biology here. And mechanism, I would say, or what's going on here that's different is not yet clear. And sort of hinting at that even with all of the sort of genetic control that you have, I would say, in these synthetic biology approaches, there's still a lot of wild biology under the hood here that that we don't understand, that the researchers don't understand. There's work ahead.
Yeah, and these changes in cell size in the non-minimal genome, they can track to this FTSZ gene.
Mm-hmm.
So it contributes to the cell size there, which as you said, makes sense because it's controlling division. Right.
Yeah, exactly. So imagine if that gene, the mutation weakens sort of the ability of the gene or the protein encoded by the gene to pinch cells in two. If you just give this a single cell more time to bathe in all of those nutrients to grow, it'll naturally be bigger by the time you pinch it, and you'll end up with, it might not at the cytokinesis, the division of the two cells from each other, it might be it'll be smaller, half the size of the original, but if it's grown bigger before that happens, it will be bigger than it would if it pinched, a few hours earlier or something like that. So that's sort of a simple sort of output there, but it looks more complicated based on that difference they see between the minimal and non-minimal cells.
So one of their ideas about the minimal bacteria unable to get much bigger, they say we've removed a lot of the membrane transport proteins, and so maybe it can't take up the resources needed to make a larger cell or maintain a larger cell under the conditions that they've used. And certainly that's a possibility, right?
Totally agree. Yeah. And I think that's where they thought, because they took away some of those membrane transport proteins, they thought that's where they might see more adaptation. That didn't happen. Yeah. And that's consistent with that hypothesis, right? That basically because they're getting by without the ability to sort of pump those in, that then, if you slow down the cell division, you won't be any bigger because you're sort of stay at this smaller size. Yeah, absolutely.
So that's their take home from that is that, so cell size is a fundamental biological property, right? Both for organisms with one cell or with many cells, and how it evolves depends on the genomic context, right? So you can see in these two organisms, it evolves very differently. In one, very slowly, probably because some genes are not there. So,
yeah, exactly. And by the way, this is like a massive, like part of the, motivation for doing this work is to try to simplify the system as much as possible to get to that minimal, set. And even then, there's enough complexity here that I think that idea is intuitive, it makes sense, but there's still like they don't test that idea, and there's that complexity even in the most simple system biologically. There's still all of this complexity or possibilities, and so think about now if we compare the life histories of whales and other marine mammals and think about just putting all of that complexity back into this. We're barely scratching the surface, which I think for me underlines the value of doing experiments like this in simplified microbial systems, where even under simple sort of relatively simple scenarios, biology is so complicated that you still there's a lot to figure out. Yeah.
I mean, and it's worth pointing out that the adaptations they see occur on unknown genes, right? A quarter of these targets that are the subject of change that Nels mentioned, they identify. They don't know what they do.
No, I love that too. And just maybe a quick note there. So the way they sort of prioritize some of those mutations is they look for some of the population genetic features. So is there an overrepresentation of mutations that change amino acids, the non-synonymous changes versus the silent ones or synonymous changes? And so, you're exactly right. And that's how they at least start to prioritize which ones to look at versus the other mutations.
I wonder if, they're missing changes in control regions, right? Because they're looking at synonymous and non-synonymous, right? So promoter changes wouldn't be looked at. And those are probably really important.
Agree. Yeah. And all kinds of ways to sort of, and I didn't get a sense, at least in the main article, how I agree with you. I don't think they addressed that. And of course there's many ways to sort of adapt. And that's one. If you have a regulatory change that has knock-on effects on, say, hundreds of genes, right? That could be a game changer in terms of some of the adaptive or beneficial situations here. Yeah. I think they kind of under considered that. But I'm not trying to cast, dispersions on this. The pretty cool result with FTZ.
No, it's neat.
Gene. Yeah,
so there are a couple of interesting conclusions. They say genome streamlining does not constrain fitness, evolution, and diversification of populations over time. You would think it might, but in this case, it doesn't look like it. And this one is really interesting. Genome minimization may even create opportunities for evolutionary exploitation of essential genes, which typically evolve more slowly. So you take out a lot of extra stuff, and now those essential genes have to change. Otherwise, the organism is going to die. So you allow that to happen, whereas normally it wouldn't because they're backups, right?
Exactly. Yeah.
That's cool.
Or there's other, I agree. There's other sort of sets of mutations that might, the trade-offs might not be as sort of stark up front. Yeah. Yep. Really cool. Yep. And of course, that's one example going from version 1 to version 3. I think you want to are some of those concepts going to hold up? But I agree, and kind of surprising that you can get maybe just again showing that the power of natural evolution, natural selection in these big populations. There's a lot of ways forward for life to go.
I have a bunch of questions here. You want to go through some of them now?
Yeah, I'd love to.
All right. So Luis has a bunch here. He says, how would it apply to viral untranslated regions? Maybe what was considered to be neutral selection actually is not so due to intrinsic cell mechanisms of defense.
Yeah, absolutely. So I think we were kind of approaching this part of the conversation just as we were talking about some of the regulatory evolution here. I think they didn't look very much at that. For bacteria and viruses, and especially as we're thinking about streamlining, what's really interesting is these things can kind of get a little bit entangled. And in some cases, some viruses even have overlapping open reading frames, which sort of throws a wrench into it. Like if you make one change that might be selected on in one of the genes, what is that? But it hurts the function of the other gene. You've got these sort of new constraints there. The UTRs are untranslated regions. I think they're, especially in streamlined genomes, sort of the definition of untranslated, and as it sort of approximates or is in the space of translated, can start to overlap. And so there can be more constraint there. This might start to help, so when we look at virus evolution and track changes over short amounts of time, it could almost seem like, if you extrapolate, if you came back a million years, there shouldn't be a single nucleotide identical between today's viruses and the ones sort of into the future. But that's not true. If we go to the past and sort of the fun field of paleovirology, where some of these virus genomes end up getting integrated into host genomes, sort of marooned there, and you can do the dating on that for millions of years. Some of these viruses like actually look almost identical to some of the contemporary ones. And so that hints at all of this constraint in the system. And so, yeah, that balancing act between, essential functions, whether it's regulatory or coding, and the ability for mutations to sort of lead to fitness benefits. Like I think what we're learning here, what we just, that answer, I think we kind of almost answered it. Like there's more possibilities than might sort of first meet the eye.
Luis quotes from the paper, furthermore, organisms with streamlined genomes have fewer targets on which positive selection can act, therefore limiting opportunities for adaptation. I wonder how this would apply to a spillover event. Well, spillover events, the ones we see anyway, they're not reduced genomes, right? So I don't think that's an issue, is it, Nels?
No, yeah, so that's right. Yeah, we're not starting out with a synthetic genome. These things have sort of been, circulating at full strength naturally, in whether it's a bat population, whatever it is. So yeah, sort of a different area. I think there's still like some definite, like the concepts here are worth thinking about. But yeah, I agree. Sort of a different question.
Tona loves the spiffy music.
I just saw Trampled by Turtles not too long ago, Vincent. They were here.
I heard. That's cool. Lake.
Yeah.
How do you match the opening jingle to the Twix? Well, this one was selected by Nels when we were putting TWiEVO together. He likes Trampled, so we used it. But the very first TWiV, right, the very first one, I just found some spiffy music online, and I liked it. And then I discovered Ronald Jenkins, and he is the author of a lot of the music on the other podcasts as well.
Cool.
So it's a little bit of a method there.
Yeah.
Mike has a question about artificial metabolic pathway that uses electricity to produce ATP from a Max Planck Society report. Could this have evolutionary implications if it's widely adopted in humans? I don't know about this. Have you seen this, Nels?
Oh, I haven't. It sounds interesting. Yeah, I'd have to kind of dig in, I think, maybe a little bit into the details to maybe give a better answer. One of the things I did just see, though, that kind of reminds me of this is new paper that just dropped on as a specific host defense is actually, when you're infected by a virus, is to start trashing your ATP to enzymatically cut it up so that the infected cell can't use that energy to produce more viruses in the overall host. And so there's no question that so I think this starts to get at, one of the core bases of evolution is the control of energy, and energy as it's the sort of potential of the energy as it's packaged into ATP, those bonds.
Yeah.
And so, yeah, evolutionary implications are, so if the trick with creating energy is sort of what you do with it, right? And so, and how metabolism is sort of shunting that to various sort of cellular processes that are going forward, which ultimately I think would read out then in, cell division or reproduction, sort of in the bigger picture, and reproductive success. And so, yeah, absolutely. As you sort of put your thumb on the scale of energy production through whatever means, I guess in this case we're using electricity somehow. I guess maybe instead of like eating plants that were used photosynthesis to gain the energy, you would just maybe give yourself an electric shock, and that would be the equivalent of eating sort of a massive salad or something. So I think all of the evolutionary sort of implications would still apply. It would just be the sort of the source of the energy would be shifted slightly. But there's plenty of, all kinds of things that are sort of at the trough and at the early steps of gaining energy or digesting food to gain energy, et cetera. And so, yeah, there could be some interesting implications there.
So Andrew wants to know, I wonder how much horizontal gene transfer factors in the wild.
Yeah, great question. And I think when it comes to bacteria, the answer is a lot. All kinds of, as you're thinking, as we bring phages into the question, like that'd be a really fun part to extend some of the synthetic biology here is, could you make a synthetic phage to go along with your a minimal phage to go along with your minimal cell? And then depending on the design, do you specifically sort of try to propagate horizontal transfer or control that, right? Like sort of kind of dial that at different levels. You could start to ask, I think, new questions about levels of horizontal transfer and what does that mean for adaptive potential, sort of at the early steps of starting from a pretty homogeneous population. Yeah, cool idea.
Yeah, horizontal is really
Cool
important. No question
sure.
about that. I wonder if you could make a minimal phage for phage therapy, Nels, if that would have any advantages.
Yeah, well, so certainly, I'd say from like an FDA perspective, one of the big implications, they're always really skeptical, or you create a high bar to get something to clinic if it's heterogeneous, because it's like, well, what are you actually giving the patient? And so the FDA becomes much more comfortable with a homogeneous population. So yeah, I don't know that how much exploration has happened there, but that could certainly be one of the applications that we're sort of talking about of bioengineering.
Priya, thank you for your contribution to science communication. We haven't seen you in a while. Hope you're okay. Welcome back.
Great to see you. Welcome. Yeah.
Les puts a link to a Trampled by Turtles video. You can go watch them.
Now we're talking.
That's cool. Happy to be part of a community which is par excellence. Thank you. Thank you very much.
Thank you.
Philip just made it from Wales. Welcome.
Welcome.
Rob, thank you for your support. We appreciate it very much. And a support again. Thank you for 2 supports. TWiEVO rules. Wow. It certainly does. Kent, the cat guy, thank you for your support of science communication. Really appreciate it. And Kent says, I've been reading A Gene's Eye View of Evolution per Nels' recommendation. What should I read next?
Ooh, good question. We did... this is a while ago. We did a TWiEVO with Daniel Kronauer, his book about ants. Sort of a different take. Like, so this isn't it gets into some of the social evolution, some of the ideas there, but just the natural biological complexity, the social complexity, with some great photos as well. I'm blanking on the exact TWiEVO number, but in the title of the book. But if you went to the website and searched on ants, that's a good one. For sure. I'll try to do a little digging on this. I think I already probably promised and haven't delivered yet, but
that would be Army Ants, right? The book from the guy who we had on, correct?
Yeah, Kronauer. Yeah. And by the way, we've also had them on. Some of the grad students from Daniel's lab have been on to talk about some of the genetic engineering they've been trying to do. That they've had some big advances recently. That's a really cool research area to bring in sort of the ability to do some experimental stuff.
So here's TWiEVO 62 Army Ants with Daniel Kronauer. And this is the book right here.
Yeah, that's it.
There it is. Army Ants, Nature's Ultimate Social Hunters. A lot of great photos in that book right now.
Yeah, agreed. Absolutely right. Yep.
So check that out.
It's a fun one. Yep.
All right. Let's see.
We also did Carl Zimmer's book. So I like this one. This is Life's Edge. We had an interview with Carl Zimmer. You cut me loose on that one for my solo debut.
Yeah, Nels was all by himself.
Oof, I was sweating it, man. But it went fine. And the book is good. Life's Edge. Carl Zimmer and I were kicking it around a little bit. And talk, like, how do you define what's alive? Actually, and that kind of has some echoes from this episode, right? As we're doing synthetic life and a search for what it means to be alive. I would recommend that one. That's off the top of my head. That's a really fun one. I enjoyed reading that. It also kind of gets into a little bit of the biotechnology as well. The basis of some of the Oxford nanopore sequencing, running DNA through pores, and how that's revolutionizing molecular biology in some interesting ways and democratizing it at the same time. It tells some of those personal stories. That's my recommendation. Life's Edge by Carl Zimmer.
Priya says, gene engineering would become a norm, then what? Well, it's already a norm in many areas. I mean, a lot of plants are engineered. I was talking yesterday to someone about, a lot of the corn is engineered to carry a toxin that kills a lot of insects.
BT toxin. Yep.
Yeah. And, it doesn't seem to, it needs to be processed by a protease, and we don't seem to do that, but it's there in much of the corn that we eat and many other things as well.
Yeah, and not without controversy, right? The whole
Yeah.
non-GMO movement, genetically modified organism. Um, and so, yeah, that's one that I've always scratched my head at as like growing up as a geneticist, I guess. And just as you learn or understand the mechanisms at play here, like you're saying, if you don't have a protease, like there's no problem. Or if you put a little bit of RNA into something that could lead to like, massive disease resistance or
Yeah.
synthesizing a vitamin, because we have an entire genome of DNA that's making RNA constantly. I think my intuition is I'm a little less freaked out about these things, and that's not to downplay it. And I'm not against conversations or understanding, but I think it's just like, that's one of the motivations of a podcast like TWiEVO, is to try to sort of have those conversations together and understand what's going on here in terms of the genetic engineering and the impacts that it may or may not have. I mean, I've even seen now, like, people are selling salt as non-GMO, and it's like, dude, since when did salt have a genome? This is sodium chloride.
I have a container of salt that says non-GMO salt.
There you go. There you go. Yeah.
I did not buy it for that property, though, Nels.
Yeah. That's right.
All right, Stefan says, I myself am curious about how pet surveillance can detect and track new variants. I don't know how. I don't think it could be, Stefan. I think the pet infection is too inefficient to be able to be a reliable indicator of any SARS-CoV-2, right? So, I think wastewater is a great way to look for new variants. Yeah, but not pets. What do you think, Nels?
No, I agree. At the same time, we've played around with, so what if we change those pets a little bit? And so the pet I'm thinking about is the zebrafish here that we put in a GFP reporter to immune responsiveness. And so there they can become sentinels. And what's fun here is actually so I agree 100%, like for real-time monitoring, making some predictions, et cetera, wastewater to me is like your best bet. But we've been curious or getting into the idea that, well, what about like predicting something? And so here I'm taking like a literal pet store example, is if I put my GFP reporter zebrafish in a tank with other zebrafish, if it lights up green, but none of those other fish look sick, that's sort of revealing some of that invisible virology in that case. And so I think that would be fun. Like, this is more like, like 20, 30 year view is as we get better at predicting or almost anticipating the immune responses that we can measure, make visible, if you can make a solid prediction on what's kind of coming down the pipeline in terms of the type of infection and the timing of it shifting from asymptomatic to symptomatic, that could give us a massive leg up, because a lot of virology, SARS-2 included, or a lot of our pandemic problems relate to the asymptomatic spread. By the time you're sick, it's too late. You've already given this to 8 people or whatever. And so, yeah, I agree with you, Vincent. Current pet surveillance, I think, falls pretty short of even wastewater and other approaches, but I wouldn't cross it off the list. And thinking about it as an active area of research is pretty fun.
I want to show you a picture of mycoplasma by David Goodsill that Nels put in the show notes. Isn't that beautiful?
Yeah, I like that. Kind of visualize through the artist's eye sort of the simple cell. This is just started. That FTSZ is assembling inside of the 2 cells there to start pinching 1 into 2.
Yep.
And you've got
Let's start to divide there. Yeah, it's very cool. Yeah. And here's another picture that I found, Nels. Look at this one. Nels in the wild.
Yeah, that's right. So we just had I was back at the Fred Hutch earlier this week. Harmeet Mullock had his 20th lab anniversary. And so we all got together. I think there were something like 70 people that showed up, Vincent. It was a big crew. And we gave short talks, caught up, had some fun meals together, hung out at the Wooden Boat Center at South Lake Union, right in the shadows of the Fred Hutch, and caught up a little bit. So that was a photo of me about to give a talk on Monday morning.
Yeah, it's sent to us by Jody.
Oh, yeah. Thanks, Jody. And I met Jody there, who is a big Twix fan. Thank you, Jody.
Yep. Yeah. And that guy who was with you, what's his first name?
That's Matt Doherty.
Matt Doherty. And he turned her on to TWiEVO originally and made her get really into it.
That's awesome.
Fields of green. Even with a minimal genome, life finds a way. It's amazing that the minimal genome survived the editing. It's impressive, but also a little frightening.
Yeah, and that's a good point here too, because I think that's like, especially when we talk about synthetic biology, genetic engineering, in this case, to like launch this bacteria. That I think it's a very natural reaction is frightening. I think for me, and maybe I was trying to get this across a little bit um, from that 2010 paper where I was a little bit underwhelmed is, the again, these like natural critters are so good at biology compared to our limited view that I think we're just making these, sort of stumbling block kind of minimal genomes where these aren't like super critters that are going to all of a sudden take over the world. um, it's sort of the opposite. You're trying to recover back to wild type levels.
Yeah.
Um, Now, at the same time, I'm not like, so inherent in all of this is biosafety, is how you think of like the kinds of genes that you don't engineer in or play around with. Even if you were trying to create a superbug, for example, a lot easier said than done. And it's again, that probability, I think, that's worth weighing in terms of what's frightening. I'm still the thing keeping me up at night. The thing that scares me are so when we think about horizontal gene transfer, when we think about bacteria, or the shuffling of antibiotic resistant plasmids and things like this, that's where I'm frightened, and a little less so
Yeah,
for
for
where
sure.
synthetic biology stands at the moment. But it's a good point. Life finds a way.
So Field says, I hadn't listened to this podcast until recent
Hmm.
office hours with Nels now starting the podcast from the beginning.
Oof,
Thank you for a thought-provoking podcast. Yeah, it's a good podcast.
awesome. Welcome.
So
Yeah.
more should listen. Mithun is here from ZA. I think that's South Africa, right?
Yeah. Is
Reed,
that,
Reed
yeah,
Africa,
8
something like that.
p.m.? Would that make sense? Yeah. 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 Welcome.
Question about orcas that were said to be speciating or evolving. Is there recent boat
Mm,
attacking behavior related to
mm.
this? Can orcas share explanatory knowledge, as David Deutsch
Yeah.
explains? I don't
Great
know about.
question here. I
I
don't
don't
know.
know.
I mean, that sounds like so. I don't think we need to speciate. So, given the sort of neuronal capacity of these marine mammals, these orca whales, these are smart creatures, and so even without invoking a speciation event,
Yeah,
which would mean sort of the populations are sort of genetically distinct either by geography, genetics, or both. I think this, you know, attacking boats falls squarely into like within species potential, I would say. Um, and but I think it's a good point to raise, which is, as our species continues to influence the environment around us, as there's more of us doing more boating, doing more of everything, um, how that relates to real-time evolution of other species or behaviors, even all of this kind of stuff. As we're, another example, if we want to go into a marine environment, as we're probably on the cusp of another massive coral bleaching event around the world, we're asking these questions, kind of doing almost these uncontrolled experiments in a sense, where I think we're hoping that there's going to be enough adaptation that coral species will go forward. But there are limits to natural adaptation, natural biology. There are limits to synthetic biology and these kind of things. And so we're kind of in a race, I think, in a sense, between the environmental perturbations that we're sort of, or seeing crop up in these sort of awkward ways, all the way from coral bleaching events to orca whales attacking boats. So yeah, we're a lot going on in this space for sure.
Peter suggests Stephen Jay Gould's essays for
It's a great,
reading.
great suggestion.
And Peter also says non-GMO corn would be grass. It's true.
Yeah, this is a great point, actually. So, um, especially as we're talking about synthetic biology here, is just kind of the efficiency of the process. So, teosinte, the ancestor of corn, and the domestication of corn, which is a fascinating story, um, involves a lot of pretty slow biosynthetic biology, which is just doing crop breeding over generations, where where breeders in really sophisticated ways are saying, okay, here's the offspring of this cross, now let's do this, and kind of getting that foothold where now you are selecting using artificial selection over long amounts of time to come up with the traits that are favorable for, like delicious kernels, a lot of biomass put into the kernels of corn, et cetera. And so what some of the genetic engineering offers, I think, is instead of that sort of sledgehammer approach, is like a scalpel. Can we just add this pesticide resistance, or can we add this great nutritional sort of property to rice or something like that? That could. So when we talk about sort of how humans are sort of with the thumb on the scale of influencing the environment, I think we depend on more efficient ways of altering some of the species that are necessary for human populations. That kind of, for me, that's where I kind of fall on the whole GMO conversation. Yeah.
Mike says, question about siRNA and COVID therapy. Has this new study been looked at? I was just looking at this. So far we just have a press release. It's in Cells and Culture, right? Treating cells and culture with siRNAs, which inhibits viral replication. So it's published today in Antiviral Research. It's still very early, Mike, and we still need to see. There are other things in development also, and it's going to be a competition to test new therapies, right?
Yeah.
So not straightforward, but we'll follow it. That's for sure.
Yeah, definitely.
Airy Ozone says, according to the piece in the Times, the orcas attacking rudders were either playing or enjoying mischief. Is there any proof of their motivation? Yeah, who knows, right?
Who knows? This is where going back to Daniel Cronauer's The Army Ants, I think makes some sense. The social evolution that you can even see in insects and starting to trace to the genetic basis is pretty fun. And yeah. That's a great active area of research, social evolution as well. Yeah.
Tona says, I just learned that apple trees are grafted to create the type of one's choice.
Yeah. And so that would be kind of one of these more sledgehammer approaches, right? Where you take rootstock, for example, graft on like a classic technique, not genetically modified, but I don't know what the right term would be.
Horizontal transfer, Nels.
There you go. I love it.
Right?
No genomes are harmed in the grafting of those trees. They all sort of are on separate areas.
All right, let's I have to leave in about 15 minutes for TWiEVO, so let's get on to some picks of the week now. So actually
Sounds good.
We have one email from Chris. You want to read that, Nels?
Yeah, let's do this one quickly, and then we'll hit picks of the week, and you've got more podcasting on the menu. So Chris writes, on the topic of mason jars, Dear Vincent, just a short note to observe that you and Nels are very skilled communicators. It's not just the simply amazing content alone, but how easy you are to listen to. No kidding. It's a real treat. No ums and ahs or superficiality. Because you're a scientist. You can see why we want to read this letter on the air, right? Yeah, of course. Scientists and very considered in what you say and how you say it. It adds to my ever grateful delight in listening. You bring the easy listening. I don't know if we're going from compliment to another category there, but by having a conversation where I feel completely welcome to listen in, you make learning fun. Same goes for all the Microbe TV mob. Many thanks and fondest regards, Chris in Toronto. Chris, thank you. That's very kind of you to say. And I think Vincent and I'll let you speak for yourself, but we totally appreciate those sentiments and are really glad that you're enjoying it. And we are too. This is so much fun to be together. I think especially in our kind of live streaming space here, where it feels like we're connecting in new ways. And so looking forward to those conversations ahead.
Yeah, I love the community here. I think it's great. And Tona says, and Vincent's a great listener. I think it's important to listen because if you listen, you're going to think of questions all the time, right? Just listen. And a lot of interviewers don't listen to the people responding, and they're just looking at their list of questions. That's not the way to do it. You have to listen, and then new questions will pop up. So if I have any good quality, it's I listen. And when you space out for whatever reason, then you lose the track, you lose the thread, right? So when there's technical issues here and I don't pay attention, you lose the thread.
No, so
really Priya says, I love Nels' backdrop. It looks like genome sequencing in distress.
Yeah, that's right. This is sort of the conspiracy theories of science brain back there. Exactly. Yeah.
Well, thank you, Chris. We appreciate the compliments. Yeah, appreciate it very much.
Yeah. All right. Well, let's move to science picks. I'll start. So I've got a quick one here, which is a great collection of stamps just dropped from the U.S. Postal Service. This is the Life Magnified stamp collection. It's a group of, I don't know, 16 or so different images under microscopes. And yeah, Life
Nice.
Magnified. Yeah,
Nice.
really great stamps. These are the Forever stamps. I think 66 each. I've ordered my first batch. There's one in the left, kind of lower left, second to bottom. It's a zebrafish. I just did that. The slide you showed at Harmeet's 20th anniversary. My second slide was actually a blow up of the zebrafish version of this stamp. And so there's also some critters, some single cell critters. I think I see Stentor in there. Some other stuff. Looking at Life Magnified with some great images. Put this on your letters if you need stamps. Yeah. How about you, Vincent? What's your science pick of the week?
All right. So we have launched a new podcast here at Microbe TV, and thank goodness I'm not involved in it. Right. So it's called Matters Microbial, and it's done by Mark Martin. And it's on episode number 3, just dropped today. And nice. Episode number 3 is with Seth Bordenstein. How much of you is microbial? And so these are basically 20-minute, this one is 28-minute interviews of Mark with different hosts.
Nice.
Kept keeping it short. So the previous one was Maya Breitbart about her work. And then the first one was with me.
Awesome.
He did a little chat with me. So I think this is a nice series. And so check it out over at microbe.tv. It's on YouTube and Spotify and Apple Podcasts and all the usual suspects. Mark is an interesting guy. Do you know Mark Martin at all, Nels?
I do. Yeah. You introduced us. So through I think the ASM, American Society for Microbiology, Communication Committee, talking about another good science communicator. I'd put Mark on that list for sure. Is he going to be doing all interviews, or what's the format? Looks like 3 great ones.
I think he wants to do short interviews with. He asked them like 3 questions basically, and he started the first 2. He would start and he would chat for 20 minutes about whatever he was interested in, and then he'd go to the interview. And I said, I think you should just do interviews.
Yeah, good call.
Let's make it focused. And so I want more short content here. Mix it up,
It's a really good idea. That's one part where I think both you and I have a lot of fun is if we have a guest on, and it's just sort of sometimes the logistics get in the way, but then we end up sort of doing our duo scenario. But yeah, that's kind of always on our menu is finding fun interviews.
Tona says Vincent's background is tidy and Nels is creative genius. That's right. Yeah, mine is this ordered thing with mostly artifacts. From. That's really By the way, I don't know. I never showed you, but when I closed my lab, I took a couple of things with me, like this spinner. Wow. Right?
Nice.
This is a spinner bottle. You put it on a stir plate, and the magnet spins, and you could grow cells in suspension. This is quite old. Yep. This is a small one. We used to use bigger ones. And then this, one of my favorite pieces of equipment, a test tube rack.
Yes.
So this is aluminum, okay, and you put little Eppendorf tubes. And now the cool thing about this is that this snaps out, and then you can clamp down the lids so you can boil it. You put some clamps on the sides, and you boil it, and the lids won't pop open. Isn't that cool?
Beautiful. Yes, it is. It's in the simplicity, the data in the kind of hints at the daily, like on the ground, on the front lines of doing experiments, having these well-designed tools can make all the difference.
Yep.
Love it.
All right. That's TWiEVO 93. You can find the show notes at microbe.tv/TWiEVO. If you enjoy our work, we'd love your support either here through the super chat, or you can go over to microbe.tv/contribute. Of course, if you have questions or comments or picks of the week, you could tell us here, or you can send them to TWiEVO at microbe.tv. You can find Nels Eldey at cellvolution.org and Early Bird. I guess it's X now, Nels, right?
On X, yeah. I don't know how much longer any of us will be hanging out there, but the uncontrolled experiment continues.
Thank you, Nels. Appreciate seeing you again.
Great to see you, Vincent.
By the way, Nels and I are going to be together in a couple of weeks. In his office. We'll do the next live stream will be from his office.
That's right. We'll be here together in LD Lab Studios. Stay tuned.
And it'll be on a Saturday, right? So unusual day.
Yep.
I will sit in front of that board of creative genius, I guess, right?
That's right. You can kick the tires here.
Yeah. I'm Vincent Racaniello. You can find me at microbe.tv. Music you hear on TWiEVO is by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology. Oh, by the way, thanks, moderators. Thanks, Andrew, and thanks Barb Mac UK, and thanks, Les. Thanks, everybody, for coming. We'll be back next month. Till then, be curious. Bye-bye.